Now we're going to look at the other really important thing that our ears do for us, and that is to help us maintain balance and equilibrium. This is a pretty complex affair, and we're going to take a fairly basic look at this. Again, here are all those structures that we looked at in the inner ear. There's your cochlea over here, and we have three loop-like structures here, and you'll notice they're kind of at right angles to one another. Kind of, I like to think of them basically like an X, Y, and Z axis here. And those three fleshy structures there are all filled with perilymph, and they are called the semicircular ducts. We're going to talk about those in just a minute. We also have in sort of the big connecting region here between the semicircular ducts and the cochlea over here, we have a large region filled with perilymph, and inside of it are two chambers, and you can see those two chambers here in the center. And those two chambers are situated, one so it's vertical and one so it's more or less horizontal. Those are called the saccule and the utricle. On the next slide, I'll tell you which one's vertical, which one's horizontal. Now, the purpose of these structures, of the semicircular ducts, and the purpose of the saccule and the utricle are to assist you in your perception of what's called static equilibrium and linear acceleration. And basically, what this all means is that when you're moving your body, like this girl is getting ready to stand upside down, or when you start moving forward in a car, straight forward, or you come to a stop very rapidly, that's linear acceleration. These structures are there to help you perceive that. We're also going to talk in a minute about how the semicircular canals help you perceive rotational movement. So let's look at the saccule and the utricle. And remember again, these are found in the vestibular area here, in this connection zone between the semicircular ducts and the cochlea. And as I said before, one sits vertical and one sits horizontal. Now, each of these guys are housed inside of a, I don't know, they kind of look like a ball filled with perilymph fluid called a macula. So you can see one macula here and you can see the other macula here. And inside those macula, your book has drawn these little, they look like little purple jelly beans. Those are the two structures where the actual perception of motion, of static equilibrium is occurring. Now, the one that's up top here is the macula utriculi. And the macula utriculi sits more or less horizontal, as you can see here. And it's really good at noticing forward and backward motion. So like when you start moving forward in a car, or walking forward or whatever, or moving back. By comparison, the one that's more or less in a vertical position here is the macula sacculi. And the macula sacculi is more about perceiving up-down motion, for example, being able to perceive when you're standing on your head like the girl is doing in this picture here. Now, if we were to blow up or look more closely inside each of these macula, you would see that that little jelly bean-like structure has within it some interesting pieces and parts. There are hair cells in there that are very similar to the hair cells that we saw in the cochlear duct. And those hair cells have at their surface those stereocilia, same as what we saw in the cochlear duct. And those stereocilia are sitting, they're bathed, if you will, in a jelly-like fluid. And that jelly-like fluid is called the otolithic membrane. Oto means ear. Lithic means relating to a rock. And indeed, if we look really closely at this jelly-like fluid, this otolithic membrane, we see in there these tiny little blue dots here are little tiny calcium carbonate rocks. Little tiny rocks made of calcium carbonate. So if somebody says you have rocks in your head, you can reply back and say, we all have rocks in our head. They're found in our macula utriculi and our macula sacculi. So these little tiny rocks are called otoliths, literally ear rock. And the purpose of them is to make that otolithic membrane, that jelly-like fluid, even heavier, so that if you move forward or back, or if you move up or down, that the heaviness of that gelatinous otolithic membrane literally pulls on or bends those hair cells as gravity pulls down on those otoliths. And it's the actual pulling or bending of those hair cells, of the stereocilia on those hair cells that we saw as before, what generates the action or the graded potentials that are needed to send signals to the brain. So saccule and utricle, those are all about allowing you to perceive linear acceleration, just when you're going forward in a car or slowing down in a car, and therefore static equilibrium, when you're standing in place, for example, or standing upside down on your head. Now, what about if you're rotating? What if you have some sort of, you're turning the corner in a car, or you're, spinning around in a circle like this gymnast is doing here? Well, then we have to employ the semicircular ducts to experience or to perceive that sensation. And when we look at the semicircular ducts here, you can see we have 3 fluid-filled chambers here, and they are filled with endolymph, not perilymph. They're filled with endolymph, and what they do is they have the ability to allow for fluid to move through them. And so if you imagine that if you start spinning, fluid is going to start moving through these 3 ducts in different speeds and different amounts. If you're spinning around in a circle, for example, then probably you're going to have a greater amount of fluid movement in this semicircular duct and not as much fluid movement in this semicircular duct. Why do we care about that? Well, because at the base of each one of these semicircular ducts is a special structure, and that special structure is shown right down here. It's called an ampulla sac, and it consists of a jelly-like substance, a little cap of jelly, sitting on top of, again, hair cells. Seen those before. And those hair cells have their little hair-like fibers that are sitting in that jelly-like medium. And then surrounding this jelly-like medium, of course, remember, is all that endolymph. So you start spinning really fast. The endolymph starts moving through this canal in particular relatively rapidly, and the faster it moves through, the more that endolymph is going to be bumping up against this jelly-like cap, which, by the way, is called the crista ampullaris. And it's going to cause that crista ampullaris to bend as the fluid pushes against it. And if the crista ampullaris bends, well, then the hair cells that are located in that jelly-like crista ampullaris are going to bend too. And when they bend, the tip links get pulled. We get the generation of a graded potential. And if I get enough graded potentials, I generate an action potential that's sent off to the temporal region, and we perceive that as a spinning sensation. And this is why even if your eyes are closed and you're in a car and it's making a sharp curve or corner, you can perceive that sensation not only because of what's happening in your semicircular ducts, but also because probably your body's getting pushed up against the car door, and you're feeling that pressure as well. If you've ever wondered why when you were a little kid, if you spun around really fast in a circle a bunch of times and you stopped, but you still felt like you were spinning, the reason is just because you stop doesn't mean the fluid, the endolymph that's present in these semicircular duct stops. As long as that fluid is still moving, and it's going to continue to move for a period of time, you're going to continue to get displacement of that crista ampullaris, and as a result, you're going to continue to feel or perceive a sensation of spinning, even though your body has physically stopped doing so. Okay, that's it on the ear. Next up, we're going to be looking at the eye.